Okay, so I'm Rosanna Milligan, and I work at I run the Seascape Ecology Lab at Nova Southeastern University. And what I'd like to talk to you guys about today is understanding behavioral drivers of diel vertical migration in mesopelagic fishes. Again, using the data from the Gulf of Mexico that we've heard about a little bit already today. So one of the most important ecosystem goods and services that's provided by the mesopelagic realm that we know of at the moment is carbon sequestration through the biological pump. We had a recent report published by Hoagland et al. At the end of last year, which estimates that about between 2 to 6 billion tons of carbon are sequestered into the deep oceans every year through the mesopelagic, with an estimated value of between 300 and 900 billion US dollars every year. That's obviously a large estimate range, and reducing some of the uncertainty regarding those values is a really important question for us. What I'm going to focus on today are the micronectonic fishes. So micronecton are organisms that are between about 20 and 200 millimeters in length. And the micronectonic fishes comprise the most abundant vertebrates on the planet, including groups that are probably quite familiar to most of us, including lanternfishes or the myctophids, things like dragonfishes that we heard about from Alex, and bristlemouths, for example. So although we know quite a lot about what's down there, we don't really have such a good understanding of the spatial temporal dynamics of these organisms, and we don't know a great deal about the behavioral variability of these guys. We know that they perform diel vertical migrations, but there is a lot of variability in here that I'm going to show you as we go through this talk, and hopefully, give you an idea of how we're starting to understand that and the drivers associated with them. So this kind of forms a main part of our work. In general, we're trying to understand what the major drivers of pelagic fish assemblages are, and we're doing this in various ways. For today, I'm just going to focus on the environmental context and these aspects of movement related to the vertical migrations. And as I said, our work's going to focus on the Gulf of Mexico. So for those who aren't aware of it, the Gulf of Mexico is a semi-enclosed basin between the Atlantic Ocean and the Caribbean Sea. And it's a really interesting case study for us. It's an ecotone between tropical and subtropical climates, and it has a very, very high diversity of mesopelagic fishes within it. It's actually one of 4 hyper-diverse regions of the world for mesopelagic fish diversity. The Gulf of Mexico was obviously also affected in 2010 by the Deepwater Horizon oil spill, which prompted extensive research efforts into better understanding the offshore pelagic realm in this basin. And that's kind of where our work fits into that. So since 2010, we've been lucky enough to be able to go and do quite extensive sampling through 2 main programs. The first of these is the Offshore Nectin Sampling and Analysis Program that was conducted over 9 months in 2011. We provided essentially 9 months of near continuous sampling across 200 by 500 kilometer area in the northern Gulf of Mexico. And we also have the Deep End Survey Program, which allowed us to go out and sample over 5 cruises in May and August from 2015 through to 2017. So we have a really extensive data set here, and it's giving us a way to kind of look in a quantitative way at some of these patterns. All our data, all the samples that I'm going to talk to you about today have been collected using a multiple opening closing net and environmental sensing system, or MOC-NES net system. And we have collected samples in a stratified way. So these we have discrete depth samples from 1,500 meters all the way up to the surface, divided into 5 depth bins. At each one of our sampling stations, we sample centered on noon and centered on midnight to allow us to kind of get a better understanding of who's living at what depths during the daytime and during the nighttime. Okay, so that's the data set we have. what I'd like to talk a little bit now about is about kind of what we expect. So in terms of diel vertical migrations, the main pattern that we see, and the one that most people are familiar with, is this idea that animals live at depth during the daytime, and then at dusk, they move up to the surface to the epipelagic waters where they can feed. And then at dusk, they move back down again to avoid predators through the day. So that's kind of the main pattern that we expect to see. But there, like I say, there is variability within this, and different organisms do this in different ways. So what I'm going to show you over the next few slides are these T-plots that you can see on the right. And just to orient you, we have depth on the y-axis, and these are our depth bins as the boxes, the bars. We have the epipelagic up here, coming through the mesopelagic, and then down to 1,500 meters at the bottom. The bars in white show the standardized abundances of fishes captured during the day, and the bars in black show the standardized abundances captured during the nighttime samples. So that's just to orient you on those plots for now. Okay, so the three kind of main groups of migrants that I want to talk a little bit more about. The first of these are the complete migrators, and this is a group where the entire population migrates every night. So the daytime distribution is at depth, and then pretty much everybody is up in the epipelagic waters at nighttime. With these guys, the implication is that the migration is hardwired and very much an intrinsic behavior for these species. It doesn't matter what's going on a day-to-day basis, they have to migrate to the surface every day and come back to depth again. This stands in contrast to non-migrators, and these are organisms where we see the same abundances basically in the daytime and the nighttime. Their vertical distributions stay the same and they don't migrate at all. That's great. Those two groups are not particularly interesting to us right now. The group that is more interesting are these partial migrators. And this is where we see only part of the population migrating at night. And this tells us that diel vertical migration is only occurring under some conditions. And the question we want to answer is why? What are those drivers? What's going on? And again, we see a lot of variability within this group. Partial migrants themselves are not a uniform group, and we see a lot of interspecific variability. These are examples from the lanternfishes, and you can see from these 6 plots, again, without worrying too much about the numbers on them, just the differences in those patterns. And what we can actually do, because we have so much data from the ONSAP and DPEN programs, we can actually start to quantify these rates among the dominant taxa and really start to get a better handle on what's going on. So the way we have been working to develop, to quantify these rates is kind of a 2-step process fundamentally. The first step is pretty simple. We simply confirm whether or not there is migratory behavior happening. And we do this with a model with an interaction term, very simple linear model. And basically what we're saying here is that the numbers caught from each depth stratum depends on whether we sample during the daytime or at night. If this interaction term comes out as significant, we know we're looking at a migrant. By contrast, we can test this with the lanternfishes, for example, which are known migrants. It works for them, so that's great. By contrast, we have the non-migrants, and this is where the model says there's no interaction term, it's just an additive term. So the numbers that we're catching at each depth bin, it doesn't matter if we're sampling at daytime or nighttime. There's basically no difference in distributions. Again, we can check this with a known non-migrator, the Cyclothones genus, and again, it works. So that's good. Once we have the migrants identified, we can then quantify the vertical migration rates using the complete sets of data. So this is quite a data-intensive method, but we take those samples where we have a complete set of 5 nets. From them, we subset the data into day and night, and from that we can establish the core daytime distribution. So where is the bulk of the population staying during the daytime? We establish that, And then we look at the nighttime samples. And this is simply a comparison. What proportion of the population is occurring in the epipelagic compared to that core daytime distribution that we identified. We do this for the dominant species, and this gives us a mean migration rate for that species across our data. So in this example, the mean migration rate is 85%. We do this for all the myctophids, and as an example, these guys all come out as migrators. This is exactly what we'd expect, but we find migration rates ranging from between 20% and 97%. So we have a mix of complete migrators in there, but there's a lot of partial migrants that we want to take a closer look at. Okay, so that's how we quantify this process with our data. What do we do with it then? Can we identify these drivers? And we know from the literature that there's a number of potential variables that might be important to us. Things like lunar phase, water mass structure, light levels through the water column are environmental variables that might be important. We also know that a number of fishes will move into deeper waters as they age. So we also have this kind of ontogenetic shift that we can consider as well. For today, I'm not going to talk about any of those, all of those things. I'm going to focus on water mass structure within the Gulf. And the two water mass types we're going to focus on are Gulf common water, which is going to be in gray, and loop current origin water, which is hot, salty water that comes into the Gulf of Mexico from the Caribbean via the loop current and provides significant mesoscale structuring within the basin. We've identified a solid way of identifying these different water mass types and discriminating between them in a paper that we published last year. Okay, so if we take the myctophid family overall, if we look at all our data, we have about 28,000 individuals that we've captured over the time series, and this comprises 57 species from 19 genera. And overall, these guys are mostly occurring in the mesopelagic. 85% of them occur in the mesopelagic during the day, and we have about a 70% migration rate overall across our samples. When we split that into Gulf common water on the left and loop current origin water on the right, they're still mostly mesopelagic in the daytime. About 85% of the individuals are occurring in the mesopelagic, but what we see in the loop current origin water, which is this Caribbean source water, is this downward shift. So most of the individuals are now occurring in the lower mesopelagic, between 600 and 1,000 meters, and far fewer in the upper mesopelagic. If we look at the nighttime migration rates, we also see a decrease in loop current origin waters from 71% to about 44%. So there seems to be this reduction in the numbers of individuals conducting diel vertical migrations. So those are the overall patterns. If we look at this on a species by species basis, and I've applied this method to almost 60 different species from Stomiiformes, Myctophiformes, a whole range of different taxa within our data set. These are the rules that hold. They apply to every species that we have tested so far, and the rules basically are, if loop current water has an effect on the vertical distributions and diel vertical migration patterns, what we see is a daytime shift to deeper waters and fewer migrants moving to the epipelagic at nighttime. Not all species care, not all species are affected. Some of them only shift their daytime distributions, some only the migratory patterns are affected. But if there is an effect, these are the rules that are always followed. So that, I think, is a really interesting finding and suggests that the water masses might be having very, very predictable effects within the fauna. One of the reasons we think that this might be happening is through light comfort zones. So we know that loop current origin waters are less turbid, and this might just simply be, they might be moving into deeper depths simply to follow that isolume within the water column potentially. What we know from previous work in terms of horizontal variability is that the myctophids in the Gulf of Mexico seem to be a well-mixed community. There's relatively little discrimination spatially within the Gulf. So we're thinking that this might be a general a finding that can be applied more widely across the Gulf, for example. So just to finish us up, key findings here, diel vertical migratory behaviours are highly variable between and within different species. The water masses seem to be having a very predictable effect on the fauna, which raises important implications for management, and also for us, for our research, raises a lot more questions. So with that, I will just say thank you to everyone who's contributed to this work, and I will be happy to take any questions.
Thank you, Rosanna.